Hello, and welcome to this week's episode of the Metabolic Classroom. I am Professor Ben Bikman, a biomedical scientist and professor of cell biology. Thanks for tuning in and joining me today as we learn more about uric acid, and in particular, its effects on the primary metabolic marker that matters most, insulin resistance. Today, we're going to be unraveling the intricacies of uric acid. So we're going to be discussing where it comes from, why it matters, in particular with a focus on metabolism or insulin resistance and inflammation, and then ultimately what to do about it. So in getting the conversation just started really clearly, let's talk about what uric acid is and then some of why it matters before we explore its origins. Uric acid is generally simply considered a byproduct of the metabolism of a molecule called purines. Now, purines are, this is some kind of deep biochemistry here, but the purines as a family of molecules are sometimes referred to as a nitrogenous base. So they have some of the chemistry that gives them a nitrogen, and they have what's called two chemical rings. You can imagine this complex structure. But when the body needs to metabolize purines, everything after all has a life cycle. It is put together and then it is broken apart. Uric acid will be a consequence of that. And we'll get into more detail on that in just a moment. But for now, just appreciate the fact that uric acid is a byproduct of the metabolism of purines. And usually when uric acid is produced, it will be flowing through the blood in the plasma, and then it will simply be expelled in the urine. The kidneys very actively will bind or grab that uric acid and move it into what will become the urine. If either the production of the uric acid is too much or the excretion of the uric acid is insufficient, we'll start to have uric acid accumulate in the blood. And once it starts to get too high, we start to get a condition that is in this earlier phase called hyperuricemia. Now, just by way of teaching you guys some of the lingo here, and I am a professor after all, hyper, of course, means that the thing is elevated. And then whenever you hear that suffix, that ending of the word emia, it means in the blood. So hyperuricemia is just high uric acid levels in the blood. And when uric acid levels get too high, it can actually start to precipitate out. So normally the uric acid is dissolved. And if you were looking at a tube of water with uric acid in it, you wouldn't see the uric acid. It's dissolved. It's mixed within the solvent of the water or the solution. However, when the levels start to get too high in the blood, the uric acid can start to come together and crystallize. And then they happen to crystallize often in joints. And so then the person can start to have gout. So gout is when the uric acid has reached a point that it is crystallizing or literally coming together as these jagged little crystals and then making people's joints ache. That is gout. But high uric acid levels can lead to higher uric acid in the urine, which can give rise to kidney stones that are sort of built on this same matrix of uric acid crystals. So that's uric acid as a general overview. Now let's get into some other specific questions as we go through this classroom episode. The first of the next questions would be us exploring, why does uric acid matter? Why am I taking the time to talk about it? And why are you taking the time to listen? Not only this is becoming an increasingly common problem, and to my delight, uric acid is becoming an increasingly scrutinized clinical marker. It seems that we hear more about it in even direct clinical settings, that uric acid is just now more and more considered just part of a normal checkup, which is really good news. It matters. So why does it matter? I already elaborated on it a little bit, namely that the uric acid will start to clump together, causing these uric acid crystal clumps, which start to literally poke and aggravate joints. Now, to lesser degree, of course, that's something that could be happening anywhere in the blood. The uric acid could be crystallizing and damaging blood vessels as well, but that's not going to hurt like a joint is going to ache. You can't feel something poking into your blood vessel. There's just no pain receptors to sense that. Now, in addition to simply the high levels of uric acid, there are additional variables that can cause the uric acid to start to crystallize and start clumping together. That can include temperature. So as temperature changes and starts to cool off, that can increase the crystallization. And that might be one of the reasons why our hands can get this, because our hands are often colder than the core of the body. Blood pH can affect it. And then, of course, as I mentioned, the concentration. Now, that is the common way of viewing uric acid, but you know me well enough to know by now that I have a metabolic view of disease. That was, in fact, the whole purpose of my book, Why We Get Sick, over one of my shoulders here, this one. That's why I wrote it. I wanted people to appreciate that among all of the chronic diseases and among all of their distinct causes, they do share some common causes. So let's get into that part of the uric acid discussion. One of the thing that interested me most over the years in uric acid is its connection to insulin resistance. Now, you have heard me discuss before these categories of causes of insulin resistance, where at a high level, I have what I consider to be primary causes of insulin resistance and secondary causes of insulin resistance. The primary causes include chronically elevated insulin, stress, and inflammation. Now, that last one I put last for a reason, because it's a good segue into the secondary causes of insulin resistance. What I consider to be some secondary causes among the definitions that I have that will qualify, or the criteria I have that will qualify something for being considered a secondary, it's that it is dependent on one of the primary variables. And that's why I put uric acid as a secondary cause of insulin resistance. Now, there is an abundance of research showing that uric acid is capable of causing insulin resistance and does so in virtually every experimental model. Increased uric acid will make isolated cells in a cell culture become insulin resistant. Elevated uric acid will make the animal become insulin resistant and the human become insulin resistant. But it does so because of its actions on inflammation. And as I have alluded to before, including very recently in my discussion in classroom about saturated fat, when inflammation is turned on, that will turn on the synthesis and the accumulation of a highly bioactive lipid or type of fat within the sphingolipid family called ceramides. And when ceramides accumulate, they will directly antagonize the insulin signal, making the cell less responsive to insulin. So if you were to start imagining a sequence of events, which I like to do and like to teach as a professor, you could imagine if we're looking at uric acid on one end, and then insulin resistance on the other, insulin resistance being the single most common health disorder worldwide and increasing the risk of virtually every chronic disease. So it is something worth focusing on. Then between the uric acid and the insulin resistance, the bridge, the great mediator, is inflammation. And then it would be ceramides, and then it would be insulin resistance. So it's worth wondering then, how important is the inflammation? And it is massively important. So uric acid will increase systemic inflammation. So inflammation is occurring throughout the body rather than inflammation coming from a wound or a general infection. It increases this systemic inflammation by activating pathways like the NLRP3 inflammasome. Now, anytime you hear that word, that suffix "ome," it means it's a kind of broad, encompassing thing. And so with inflammasome, it's basically referring to this NLRP3. You could imagine it as this big central switch that when it's turned on, power throughout all of these other circuits is being turned on. So when NLRP3 is turned on, it's activating countless other inflammatory signals. So it's really at a nexus that when it gets activated, a lot of things will follow, or a lot of things will be turned on as a consequence of that one. And again, uric acid will turn that on. And then there was a good study by Zhu, Z-H-U et al., Zhu et al. In 2014, and we'll have this link in the show notes. that really explored this and found that uric acid was capable of very rapidly causing insulin resistance. And then there was a substantial inflammatory burden that was a part of that response. And indeed, I would say an essential part of the response. That if you, to be very precise about it, if you block the inflammation, you block the insulin resistance. So in other words, uric acid is only causing insulin resistance in the cells or the body because of its effect on inflammation. So that's an effect. In fact, I'll get to some of my own research in a moment that we're about to publish, where I can solidify some of that signal and that sequence of events. All right. Now, where does uric acid come from? At this point, you understand generally what uric acid is, this molecule in the blood that has the potential to both crystallize together, causing gout, or just activate inflammation signals. You have a better understanding of why that matters in the context of insulin resistance, which we're going to revisit when we talk about solutions to this. But then let's move on now to discussing, where does uric acid come from? I mentioned this right early on, describing or mentioning the origins coming from the purines. So as I said, as a term of chemistry, so pardon the terms getting a little technical, but purines are what's called nitrogenous bases that are used as the building blocks of nucleic acids like DNA and RNA. So, of course, you've heard of DNA. When you start to think about the genetics of every cell on the planet, purines are going to be a part of this. So every animal, every plant, the purines are an essential component within the nucleic acids that make up all of our DNA and then the central dogma of cell biology. So the idea of what makes our genetics become our body, that flow of genetic information, boy, the purines and the nucleic acids are just critical to that process. Now, why or how can this become a problem? The conventional thinking with uric acid, still conventional and certainly historical, is that foods that are high in purines, where you're eating a lot of purines, require the body to metabolize and clear a lot of purines, and thus giving rise to more uric acid, which through a pathway I'll get into in just a moment. But foods like this particularly results in the vilification of things like red meat and seafood. Red meat and fish tend to have a higher purine load. And thus the idea is that if you eat more of those purine-rich foods, your body has to deal with them as a course of breaking it down. If you have more purines than you need, than the body needs, you're breaking them down. So when you start to break down the purines, then it gives rise to a molecule called xanthine. And as xanthine is metabolized, as you continue to clear these molecules and break them down, at the end of it then, as xanthine is acted on by its enzyme, it turns into uric acid. That's one of the products. So it is, you eat it, you metabolize it, and it gets turned into uric acid. That can certainly happen. Now, the conventional view is that is not only the conventional view is that is the only thing that happens, but also that it is certainly the most important, even if someone is considering that there might be another cause. But now let's talk about that other cause, because it's the one I believe is the more important one, which is fructose. Some of the evidence that I would cite just at a very superficial level to support the idea that if we're worried about the increasing frequency of gout, and it is more frequent, then how do we square that curve? How do we reconcile that with the fact that red meat consumption has been going down generally, and fish consumption has always been really low? Well, then how do we reconcile that? If one of these variables is going down, or at the best staying flat, how can the other one be going up? There must be something else that explains it. And indeed, I think that something else is fructose, the consumption of which has been increasing for decades. Now let's talk about fructose then. So fructose is absorbed in the intestine, and it is moved into the body, and it goes to the liver. And the liver is uniquely suited to metabolize fructose because of the transporters that allow it to bring in the fructose, and as well as the enzyme expression enabling it to break it down. So unlike glucose, when you have these monosaccharides, these simplest unit of the sugar, glucose and fructose, when they come into a cell, they each have their own sort of respective pathway that they're going to go down. Fructose, in order to be metabolized, glucose is heavily regulated. These steps are intricately sensitive and responsive to the overall energetic milieu of the cell. So if the cell has a lot of energy, let's think about the liver. If the liver cell has a lot of energy, it can take that where it's burning a lot of glucose already. That high level of glucose burning will basically start to feed back and tell the liver cell, hey, we're burning a lot of glucose right now. So let's divert that glucose into something else. So it has some regulatory steps that will check its own metabolism. However, fructose bypasses the main regulatory step of glucose, which is an enzyme called phosphofructokinase, PFK. Now, PFK has some different varieties, but PFK, phosphofructokinase, is one of the more sensitive steps in the burning of glucose that allows the cell to decide what it wants to do with glucose. But again, fructose bypasses that. It doesn't have to listen to the signal coming from phosphofructokinase like glucose does. This means that the liver cell is able to metabolize fructose at a much, much higher rate, essentially unchecked compared to glucose levels. Now, then within the liver, as the fructose is getting metabolized, it's getting acted on by an enzyme called fructokinase, or sometimes called ketohexokinase. Now, that has nothing to do with ketone bodies, but rather it's more of a term of specific chemistry where there's a ketone bond within these molecules. So let's just call it fructokinase to avoid any potential confusion. So this fructokinase enzyme turns the fructose into a molecule called fructose 1-phosphate. But whenever you have a molecule that has gone from one form to a phosphate form or a phosphorylated form, it has to have used ATP in order to do that. So fructokinase will grab the fructose molecule, it will grab an ATP molecule and mesh them together, one of the phosphates at least from the ATP, and give rise to fructose 1-phosphate. But that means that what was an ATP with 3 phosphates, adenosine triphosphate, has become broken down, having lost 1 phosphate, and now it's adenosine diphosphate, ADP. And this reaction just keeps happening. We keep just breaking ATP just to handle the fructose load because we were metabolizing so much fructose, and essentially the liver can't stop. There's no way for it to regulate its own fructose burning. The net effect of this is that as we start to try to recreate ATP in order to continue to fuel the fructokinase reaction, we end up getting a lot of a molecule called AMP, adenosine monophosphate, AMP. That is an important development in this process. When we start to get a lot of AMP, now we're just a couple steps away from the uric acid, because AMP starts to turn through a series of events, and I'll spare you. You're already thinking I'm getting too specific. I could be a lot more specific. Where the AMP is now basically breaking down through a few steps back into xanthine, which you recall because I just mentioned it, and then give xanthine another step and it's turned into uric acid. So we've now completed this journey. Again, where the fructose, just to recount it, perhaps with some degree of brevity, the liver can't stop burning fructose. There's no way to really turn it off, unlike other metabolic pathways. And this means that the liver cell is burning through a lot of ATP. And through the course of trying to continue to recreate ATP, we end up getting a lot of AMP. And then the AMP, after a handful of metabolic steps, becomes uric acid. This, I think, is the singular pathway mattering more than any other when it comes to explaining why the liver cell in particular is creating so much uric acid. Like gangbusters, it can't stop. So the more uric acid coming in the mouth, or the more fructose coming in the mouth in whatever form, whether it's fruit juice or sugar or high fructose corn syrup, is going to give rise to more uric acid production by necessity. All those molecules of fructose you just can't stop burning gives rise to a lot of uric acid. Now, before I move on to the final part, I need to encourage all of you, if this is a topic that you're getting more and more interested in, I strongly recommend you look up the work of Dr. Rick Johnson, a friend and colleague in research and collaborator that I'll share the project we're doing in a moment. But he has written a book called The Fat Switch, or Nature Wants Us to Be Fat. And it's all about how then the uric acid, as it accumulates, starts to drive more hunger and then it ends up becoming a primary contributor to obesity in the long term. Now, with all of this having been said, we move on to the happy ending, which is what can you do about it? Thankfully, you can do a lot about it. Let's start with the pharmacological approach, and then we'll move into the nutritional approach, which you can already guess where that's going to go. The pharmacological approaches include very common drugs like allopurinol. Allopurinol is the most common, and it will effectively reduce uric acid. In so doing, it will improve gout, as is expected, and that's usually what people are looking for. They want resolution of their gout, that being the most commonly acknowledged consequence of high uric acid. But there are metabolic effects as well that are beneficial. So a study by Madero et al., 2015, Madero et al., they published a clinical trial. So this is good. That's the highest quality of interventional research. And they looked at overweight and pre-hypertensive subjects. So these are men and women. And they put them on allopurinol. And they found that the allopurinol not only resulted in uric acid reductions, but also a significant improvement in metabolic health. Now, a confounding variable, including weight loss and improvements in insulin sensitivity. So all very, very good things. One challenge, though, is that I have been unable to find a study that did this in the absence of any other intervention. So this study by Madero et al., 2015, they gave the people allopurinol and looked at these metabolic outcomes, but they also had a low fructose diet. So based on what I just said, you can see how that's a bit of a confounding variable. Even still, generally, certainly in clinical practice, from what I've gathered from clinical teams, allopurinol will often result in some degree of weight loss and general improvements in metabolic markers that are connected to insulin resistance. Now, however, before you just jump into taking allopurinol, every drug has side effects that you don't want. And so I just feel inclined to mention them just to curb your enthusiasm for this drug, lest you ask for a prescription from your clinician a little too readily. There can be some significant skin reactions, like an exfoliative dermatitis, as well as some even more serious skin problems. There is the common GI issues like diarrhea and nausea, and even some liver and kidney problems as well. So it is a drug to be careful with, like all drugs are, even though it does have some metabolic benefits because of the lowering of uric acid. Now, let's move into allulose, as I mentioned at the beginning. Allulose is a rare sugar, and it really has emerged as a pretty promising agent to improve uric acid, to reduce uric acid. And that might be one of the mechanisms whereby allulose has been shown so extensively in humans and rodents to improve metabolic outcomes. Now, just as a disclosure, I am finishing an allulose study now. We're just about to submit this for publication in animals, and we're doing a human study soon. But the effects were substantial. And these are unpublished results that I'll mention, so I need to describe them cautiously, or just with some, you need to hear them cautiously because they've not gone through a peer review process. But among the groups we had was one group of animals on a Western diet, so high fat, high sugar, and unexpectedly they gained a lot of weight. If we gave them allulose in their drinking water, they gained substantially less weight, less than half of what the other animals ate, and they ate less. They just were not as hungry. They had much better satiety. So allulose is important. It matters. And uric acid could be one of the key changes that's helping the metabolic health of the animal. Now, how does this happen? It could be twofold. One with regards to absorption, and the other with regards to excretion of uric acid. Now, let me also say, I regularly get my blood tested through a company called Blokes, and I encourage you to look them up. I really am a fan of their service. My uric acid levels have always been in the normal range. However, when I, as just a matter of experiment, started taking much more of the RX sugar products of the allulose, my uric acid levels went below the normal range and nothing really changed, but they went down so much that it got below the normal range. I don't think that's a problem. I've continued to feel fine, but I've seen this happen in many other people as well, who, despite changing their diet and becoming healthier, they still have persistently high uric acid levels, and it just concerns them. They start taking allulose and it drops very, very quickly. So now I appreciate what I'm saying. These are unpublished results, so please appreciate that. I want you to appreciate that as a scientist. What I'm citing to you or sharing with you is anecdotal, my own and those I've seen of others. But I have seen enough that I am inclined to mention it, even though I would rather just be citing a peer-reviewed study. Until we have some peer-reviewed studies, I'm happy to mention the anecdote. Now, how can it work? All right, the first thing I mentioned was the uric acid. Interestingly, allulose is the good twin to fructose. Now, and a lot of people nowadays are trying to say fructose is good and you should eat a lot of it. I'm not trying to suggest that fructose is uniformly bad, but I do think we eat it in a way that we never did before. Honey would have been a very uncommon occurrence, and we would have had to fight off bees in order to run away from it. So it's something we as humans would have enjoyed infrequently. And moreover, the fruit that we would have had ancestrally would have not only would not have been drinking it, we would have been eating it, but also would have had very, very low levels of fructose. Just as a case in point, do yourself a favor as an educational tool and look up the change in apples over the past 100 years. What was once these small, bitter, sour little things have become these big, fat, juicy, and delicious fructose sugar bombs. So we eat a lot of fructose. Now, back to allulose. Allulose is a twin. It has one singular alteration in one of the carbons, but compared to fructose, but it is otherwise identical to fructose, but it is different enough that it can't get metabolized. But it still can compete with those same absorption pathways. So the same doorway that lets fructose come in can let allulose come in. And so that, we believe, is most especially relevant at the gut. That if you have competition for the uptake of any fructose, allulose is fighting now. What used to be an uncompeted entrance for fructose to just come right into the gut now has to fight. And with the allulose, if you're taking an allulose, so it stands to reason that less fructose is getting absorbed. That means less has to get metabolized. Now, let's say that there is fructose that has been absorbed and it is getting metabolized. That now we look at the back end of it, where uric acid, if there's more allulose, this is theoretical, this hasn't been published, but we're hoping to confirm this in our upcoming human study. When allulose gets consumed, it's because we don't burn it as an energy source, its main exit is through the urine. And there's this idea that Dr. Rick Johnson at University of Colorado, the global expert on fructose and uric acid metabolism, he has posited this idea that the allulose could be increasing this pull effect on the uric acid, enhancing the kidney's ability to remove the uric acid from the blood and dumping it into the urine. And I already shared with you my own experience and that of others. Now let's transition as a final point into the ketogenic diet. The ketogenic diet is calorie for calorie, probably the best way to improve insulin sensitivity, and I would even say weight loss in the body. Interestingly, a ketogenic diet often, but not surprisingly, will be high in meat, including red meat and even fish. So those are foods that are high in purines. Now, typically, this person's not eating a lot of fructose, of course. That would be antithetical to the ketogenic diet. And as we mentioned before, when purine metabolism is high, uric acid production goes up. Now, there are studies to show that uric acid levels can stay high or even climb with the adoption of a ketogenic diet. This is documented, it's published, and I have no qualms in sharing that. So how do we reconcile that with the fact that I just mentioned how I believe, based on the evidence, that calorie for calorie, the ketogenic diet's the best way to improve insulin sensitivity? Well, we just got done talking about how uric acid will increase inflammation and thereby cause insulin resistance. Interestingly, the ketogenic diet, even if some of the studies show an increase in uric acid levels, uniformly show a reduction in inflammatory signals. So when you look at something like C-reactive protein, it is always down in the group on a ketogenic diet. And yet sometimes uric acid is up. So what is going on? How do we reconcile all of this? The reconciliation, I believe, is explained in the study that we've just finished with Dr. Johnson as a collaborator. The Dr. Johnson I've alluded to a couple times now. These are once again unpublished. I have, at this point in time recording this podcast, a handful. I think I have 3 studies that I need to finish and get published or submitted for review to publish. This is one of them. We wanted to answer this question, and so we conducted a series of cell culture-based experiments. So the simplest model, right to cells growing in petri dishes, where we found if the cells were treated with uric acid, inflammation was up and insulin resistance occurred. When we treated with uric acid and an anti-inflammatory agent, insulin resistance was better. When we treated even further, one more step, with uric acid and an inhibitor of ceramide production, the cells were insulin sensitive. So you could see that we addressed it at each of these subsequent steps. Remember the pathway that I outlined earlier. Uric acid causing inflammation, causing ceramide accrual, causing insulin resistance. If we increase the uric acid, we get insulin resistance. If we increase the uric acid but block the inflammation, the cells are insulin sensitive. If we increase uric acid but block the ceramide production, the cells are insulin sensitive. Now, however, where do the ketones come in? Earlier on, I had mentioned that uric acid activates the NLRP3 inflammasome. Well, guess who inhibits the NLRP3 inflammasome? Ketones. And so it could be that in the ketogenic diet, despite the increase in uric acid, the ketones are sufficient in order to keep the consequences of the uric acid in check. Now, as an interesting next step here, that one of the possible explanations for why a ketogenic diet can increase uric acid may be the high consumption of meat. It may also be the fact that when ketones are getting excreted in the urine at the kidney, they actually compete with uric acid. And so if you have more ketones in the blood, it could be not that you're making more uric acid, but just the very act of clearing the ketones means the uric acid levels have to wait a little bit. The uric acid molecules need to wait until they can get in line to finally be excreted, potentially giving rise to the higher uric acid levels. You could look up an old study published in 1965 by Lecoq, L-E-C-O-C-Q, et al. Lecoq, 1965, L-E-C-O-C-Q. We'll put this in the show notes like all of them. But you can look that up there, and they detail this pathway of this competition for excretion. Anyway, that could be a part of it. But again, the evidence does suggest that if you are on a ketogenic diet, you don't need to be as concerned about the uric acid because you're also making molecules that are inhibiting the inflammation. That's it. That's today's metabolic classroom. I hope by this point now you feel much more confident conversing about uric acid, what it is, why it matters, where it comes from, and even what to do about it. Thanks for tuning in. Thanks for listening. As ever, nothing thrills a professor more than having a new bunch of students to teach a principle to. Thanks for joining me.